back to acids and bases. We talked a little bit about what acids were, what bases were. Acids give up hydrogens. Bases take hydrogens. Bases actually, they take hydrogens because they have hydroxide ions present. Okay? The hydroxide ion, which is this guy right here, can bond with the hydrogen to make H2O, to make water, so they neutralize each other. All right, and we talked about we mentioned the pH scale. Remember what it stands for? The P stands for what? Close. Fancier word. Potential hydrogen, right. Okay, now, someone tell me the difference between strong acids and weak acids. Do you remember? What about dissociation? Okay, strong ones completely dissociate. Weak acids don't dissociate as much. Okay, that's going to come into play a little bit later because we're going to come back to what's called carbonic acid. Remember that from the respiratory system? We're going to come back to carbonic acid, and it is actually a weak acid. It does not fully dissociate. So we've got a little bit of math involved with that.
So why, when you put the sodium bicarbonate and the carbonic acid together?
Okay.
So why does that make a good? Why cannot one be good as the other?
I'll show you. I'll show you. Why do you have to have both instead of just one or the other? I'll show you. Okay. Now, pH was potential hydrogen, and I showed you the whole exponent thing and why the numbers are in reverse. Okay. Anything with, here's the pH scale. It ranges from 0 to 14. All right. 7 being in the midpoint, that represents neutrality. That represents sort of equal amounts of hydrogen ions and hydroxide ions present. No lopsidedness. It's sort of like what pure water would be. Now, the farther you dip below 7, and the farther you get away from 7, okay, and go towards 0, the more acidic this becomes. The farther you go towards 14 in this direction, the more basic it becomes. Or another word is alkaline. The more alkaline it becomes. Okay? So know those two words are the same. Now, as far as blood, you will find that blood's anywhere, it's slightly basic. It's about 7.35 to 7.45 in terms of its range. So a little bit basic because of mostly the things that's dissolved in it, especially all that bicarb and other things that are in there. Okay, now, how do you calculate pH? You're going to have to know how to calculate the pH. The pH is just a logarithmic scale. Okay, each step is a factor of 10. It's the easiest and most efficient way to keep track of our hydrogen ions. For example, something that is a pH of 6 versus a pH of 5. Something that is a pH of 5 is actually 10 times more acidic than something that's a pH of 6. Something that's a pH of 4 is actually 10 times more acidic than something that's a pH of 5. Something that's a pH of 4 is actually 100 times more acidic than something that's a pH of 6. Each step you go, you multiply by 10 in terms of the factor by which it is more acidic. So if you go from 6 down to 3, you go 10 to 5, times 10 to 4, that's 100, times 10 more to 3, that's 1,000. So something that's a pH of 3 is actually 1,000 times more acidic than something that's a pH of 6. So you see how we can cram a lot of range and a lot of information using some small, simple numbers. Okay? So my point of this is even the tiniest little pH change on the scale could represent a huge change in terms of hydrogen ions in the body. Okay? So just because your pH drops from 7.35 to 7.3, realize that that's not insignificant. That is actually significant, even though it's a decimal change. Okay. Now, how we calculate this, see this right here. It says pH, potential hydrogen, is equal to the negative log of the hydrogen ion concentration. These brackets means concentration. That means a number that represents how many hydrogens are there. Now, to convert that concentration like when we did our diffusion osmosis lab, like 0.2 molar sucrose, that was a concentration. You'll take a concentration value, you'll take the log of that value and put a negative sign with it, and you'll get the pH. Now, you don't have to understand all of that, and you don't even have to know how to use your calculator on it because I'll give you the stuff that you need on the test, okay, to calculate this. But if you were doing it on a calculator, you would type in, let's say your hydrogen ion concentration was 0.001. You would type in 0.001, hit the log button, and when you do, a number will pop up on your screen, and it'll be a negative number. The reason this negative sign is there is to tell you to take the negative of a negative. So turn that number into a positive value, and that's going to be your pH. Okay? So all this logarithm means like a factor of 10, for example. Like the whole thing we just talked about, 10 times 10 times 10 with every step. That's a logarithmic scale. So that's how you convert. What I will do for you is eliminate the math, and I'll just give you, if I ask a question on the exam, I'll say the log of this is this. The log of this is this. I'll give you those values, and you pick which one you need to use for the problem that you're solving. Okay? You don't have to worry about calculating punching this in the calculator, doing the math, or anything like that. It'll be there. Okay, but no, that's what it is. All right. In the body, acidosis and alkalosis. Acidosis means increased acidic conditions in the body. Now, it's a relative term. Notice what acidosis, where it actually starts in the human body. It's generally accepted to be anywhere below 7.35. Now, realize that 7.35 is actually slightly basic, right? It's not actually in the acid part of the spectrum of the pH scale, but it's still a pH below what's normal. So that's considered acidosis. pH of 7 is actually neutral, but that's actually a lower pH than what blood should be. So they call it acidosis. If you get above 7.45, that's alkalosis. There's too much of a basic environment, so a lack of hydrogen, for example. All right, now we talked about dissociation, strong acids. If you look up here, you'll see what happens when you take hydrochloric acid and put it into solution. You get hydrogen ions and chloride ions all floating around completely separate from one another. That's a strong acid. Carbonic acid. If you could freeze time and take a look into a beaker of containing carbonic acid, what you would see is some of them would be separated, like you see here, but there would be a lot more that are actually held together, like you see here. It's not completely dissociated at any given time. They tend to stay together a little bit more. Okay, now, if our hydrogen ion concentrations get screwed up, what kind of effects does that take on the body? It says it can alter nerve, enzyme, and potassium ion activity. Enzymes, for example, we know that enzymes are extremely important in biological processes. They're proteins that are not consumed up in a reaction but help reactions take place. Enzymes can get denatured with hydrogen ions. What denatured is, if you remember back to basic anatomy, you probably learned a little bit about enzymes. If not, I know in micro you covered a little bit about like primary, secondary, tertiary structures of enzymes and things like that. Basically, an enzyme is a protein, and then it gets all twisted up and wadded up and sort of bundled together, and now it's floating around as a chemical that we call an enzyme, a big one, big chemical. Now, what happens is it's all folded up, sort of like a wadded up phone cord or something. It's all folded up and stuck together, but the paths that are right next to each other can actually kind of stick together and hold the shape together. Okay? These bonds, these disulfide bonds, sulfur is the atom that's responsible for holding these together. That's why whenever meat and proteins fall apart and it rots, you get that sulfur type of smell. But the proteins are being held together, and then what hydrogen can do is come in and sort of start reacting and breaking those bonds and denaturing and rendering those proteins and those enzymes useless now, and then thus life cannot continue if that gets too high. Okay, if there's too much destruction of these enzymes. Acidosis can certainly depress the nervous system. It makes it not as responsive. Alkalosis, the opposite of that, a deficiency of hydrogen or an excess of hydroxide. Alkalosis can cause overexcitability of the nervous system. So they work in opposite directions, but still just as detrimental. I talked about this. You can affect enzyme structure. How can you get extra hydrogens in the body? Because we just don't go around drinking acid, okay? Well, you sort of do if you drink a lot of pop, but or juice. But where you're going to get a lot of these hydrogens is from just metabolism, going through cellular respiration, going through all that Krebs cycle stuff and everything like that. So metabolic activity can put hydrogen ions back into the ECF. Carbonic acid. Whenever you form that in your body from generating carbon dioxide and reacting with water, that creates hydrogens that can be released into the body. When you break some nutrients down, the byproduct sometimes are acids that float around in the bloodstream until they're dealt with. just different steps in the sequence of biochemical events that occur in your body. You're constantly producing these byproducts that are acidic in nature. Now, what does our body do to oppose acidosis or alkalosis? There are three big things that we're going to discuss, how we can help control this. The first one is very generic. It's called chemical buffers. Buffers have the ability to help control hydrogen and hydroxide concentrations of ions. Okay? If there's too much hydrogen, for example, a buffer system can absorb some of it, pull it out of solution, and lock it up. If there's not enough of it, it can take that which it has locked up and free it out into the solution. This is what buffer systems can do. The first one is called chemical buffers. These are going to be chemicals that are just floating around in the blood, traveling through the body, kind of like a generic immunity when we talked about all of that. This is sort of generic chemicals that are moving around throughout the body that have the ability to bond and release hydrogen and help take them up or release them as needed. There's also respiratory mechanisms. Realize that by controlling your carbon dioxide, you can control acid levels. The more carbon dioxide you retain and keep in your body, so if you hypoventilate, bring more carbon dioxide in, what you do is bring more CO2 that can react with the water and thus produce more carbonic acid. So if you need to increase your acid levels, if you're experiencing alkalosis, one of the things it could do is you want to go from alkalosis back towards acidosis. That's where you want to go with that. So you can slow down the breathing. You can retain more carbon dioxide. That'll increase your acid levels. Okay? If you want to go the opposite direction, you'll have to hyperventilate, get rid of a bunch of carbon dioxide. Okay? That's by blowing out a bunch of CO2, you reduce the amount that can form with water to form carbonic acid in the body. So the respiratory system has a very important role in that. Renal mechanisms are more precise. We talked about the kidneys and how they can actually pull in hydrogen or from the into the tubule from the blood vessel, or vice versa. They can put it back into the blood. They more finely tune whatever control. They got better control than the other things do. But those all three can work together. Just to give you an idea of the pH scale, some things that are on there. Our gastric juices in our stomach has a very low pH. It's very, very acidic. Here's a lot of other things, and then some basic kinds of things over here. Why do you take milk of magnesia to settle an upset stomach, right? I mean, the whole point is to, like heartburn especially. Heartburn is not really anything to do with the heart at all. It's acid reflux types of situations. It comes up past the gastroesophageal sphincter and kind of get into the esophagus where there's not that thick mucus lining like there is in the stomach and that kind of thing. So to coat some of this, what you want to do is neutralize acid to help knock down those levels a little bit. Use a base. Use something that will react with acid to just produce something neutral like water, like some kind of salt water. Okay? All right. Like I said, between about 7.35 and 7.45 is the average range that you're going to find the blood in the body. The higher pH, the more basic you're going to find in the arterial blood, the more acidic in the venous blood. Why? Why would it be a little bit more acidic in venous blood? Exactly. You're collecting carbon dioxide from the tissues, so it's going to be a little bit more acidic. Okay, good. Okay, a buffer system. How does it work? There's two things. There has to be a pair of substances. Okay, and one of the most important in the body is the carbonic acid carbonic acid bicarbonate buffer system. Okay, that's ours. One of these substances can form free hydrogen and release it if necessary. The other one can absorb if necessary. Okay, so if you look over here, and you have let's say you have a solution of salt water. Okay, sodium ions, chloride ions floating around. It's unbuffered. Okay, that means if you drop hydrochloric acid in it, that's what you're going to get. You're going to get not only sodium and chloride dissolved around there now, but you're actually going to have hydrogen ions and chloride ions all floating around. Very high dissociation. Hydrochloric acid is a strong acid. There's nothing there that can grab onto that hydrogen. Now think about it. Why? This makes sense. Okay, we know that opposites attract, right? Positive charges, negative charges come together. We also know that this ion right here is a chloride ion, right? That's the same ion that you're bringing in up here. You know, by virtue of the fact that hydrochloric acid is a strong acid, the second it hits that solution, does hydrogen have a good affinity, a good attraction to chloride? No. The second it hits that water, it splits apart, right? So it's not going to make any sense that chloride ions here would have the ability to absorb hydrogen out of the solution. They have a very loose affinity to it. They don't want to stick to it. Otherwise, you wouldn't have had this strong acid happen in the first place. Does that make sense? You see what I'm saying? If you drop this in, this chloride ion is the same as this one. The degree of attraction between that hydrogen and that chloride is actually very weak. That's what makes it such a strong acid. It's a weak attraction, but it means a strong acid because when you put it in water, it means it's going to fall apart very easily. Okay? Now, if that's the case, when I put it in water and it falls apart very easily, what a buffer would do is start reacting with those hydrogens and pull them in and stop making them free, lock them up inside of a chemical, inside of a bond, so that they're not freely floating around by themselves. Chloride doesn't have the ability to do that. That's the only thing in here that could possibly stick to that hydrogen, right? It's the only negatively charged thing in this container. So the only thing that you can stick to that hydrogen is that chloride ion. But we know that chloride doesn't stick to hydrogen because hydrochloric acid just dissociated. Okay? So this is not a buffer system. It will not work. It will not allow you to absorb necessary hydrogens and then release it later. Okay? Now, take a look at this, though. You've got H2CO3, which is carbonic acid. Notice they're still intact, a lot of them. That's a weak acid. Okay? You've also got bicarbonate right here. This is a weak base as well. So you have these two things together. You'll notice that the bicarb ions are just floating around as charged ions. Some of the acids are stuck together. What happens now when you add in hydrochloric acid? If I dropped hydrochloric acid, note why this Na is here, just so you know. Sodium bicarbonate. That's actually baking soda. Okay. Sodium bicarbonate, you had to have dissolved that in the water, and when it dissolved, these ions separated from each other. Okay, so the sodium separated from the bicarb. Now this sodium is just floating around. It doesn't mean squat. It's not going to do anything for us. It's just it's actually called a spectator ion because it's watching what's going on. What we care about are these two guys right here that are present. Okay, now what happens whenever we add, let's say 3 HCl molecules, 3 strong acids, molecules to this mix? All right, now look at everything that's going on. These red things are the chloride ions. Again, that's going to be a spectator ion. We know that it stays dissociated when that happens. So you've got your spectators here, sodium, you've got spectators here. The key, the most important thing here is how we deal with the hydrogen. We've got carbonic acid molecules right here. We've got hydrogen right here. And we've got some free bicarb over here. What has happened is notice that, see this guy right over here? This bicarb, another bicarb, looks like maybe 2, I don't know. 2 bicarb, there was 3 up here. So these 2 bicarbs right here. What they were able to do is, since they are negatively charged, they want to accept hydrogens. They can accept hydrogens if hydrogens are put into that solution. Okay? Unlike what chlorine can. You're going to have to go with me on this because I don't have time to explain all the chemistry behind it. Okay? But just realize that bicarb has the ability to stick to hydrogen even though chloride can't. So what you'll get is when you put these hydrogens in and they separate out, one of the hydrogens floated over here and stuck to this. It made this guy right here. Okay? Made this guy right here. One of the other hydrogens floated in and stuck with this one. Made this guy right here. Okay? Now what it's showing you also is one of them didn't, but if you give it long enough, it'll probably will. It's just what happened here is one of them didn't. Here's a hydrogen floating here. Here's another bicarb floating right here. They have the potential to be able to come together. Okay? Now what just happened is this could represent a buffer situation, and the reason is because you added acid, and remember, to affect the pH of something, what you have to do is alter the amount of free hydrogens floating around in the solution. If you put in, like up here, if this was just salt water and you put in a bunch of free hydrogens, you just plummeted that pH. It went very acidic and very, very, very quickly. Here, you put that same amount of hydrochloric acid in, but look what happened. Here you had 1, 2, 3 hydrogens floating around. Here you've only got 1. You've virtually not affected the pH, or at least affected it very minimally in a buffered situation because there was something there that could absorb that hydrogen. Now at the same time, what you got to realize is carbonic acid is a very weak acid. That means it doesn't dissociate very well. So it kind of stays locked up and can hold that hydrogen. But that doesn't mean it is incapable of donating that hydrogen. It is still an acid. It still has the ability to release a hydrogen. So you had to have both present. Now you have this carbonic acid here. If you need more free hydrogens to be in your solution, it'll release some. And some of this hydrogen can be released and put back in if you need to alter that. So you've got both of them present. Carbonic acid is necessary so that it can give off hydrogens if it needs to. Bicarb is necessary so that it can gain hydrogens if it needs to. Okay? And the reason that it all works together is because it's a weak acid. It doesn't dissociate completely. And because it doesn't dissociate completely, you're going to need some extra ones of these in here. If you relied solely, okay, think about this. I don't know if this will make sense. I'm trying not to confuse you, but at the same time, I'm trying to sum up a whole bunch of chemistry in a very short amount of time. Take this, for example. Take this carbonic acid. If you wanted to use only that and not put any of this in there, okay? If you just wanted to use carbonic acid, you would have to realize that you need it to do 2 things. You need it to be able to fall apart to make this, and you need to be able to make it stick together. Okay? This actually sticks together much easier than it does fall apart because it's a weak acid. It likes to stay together more than it likes to stay apart. So if it is going to have the ability to absorb hydrogen, it's going to have to fall apart. Right? If you want this to have the ability to absorb hydrogen, you're going to have to give it a jump start. You're going to have to put more of these in to start with because this doesn't dissociate very well. It doesn't fall apart and make these very well. So you need to bring something else in like this that's connected to it that does fall apart very easily and give yourself a leg up. Give yourself some more of these present right at the very beginning. Okay? All right, if you don't get all that, that's fine. You don't have to. But what you do have to understand is that this makes a buffer system. Carbonic acid, bicarbonate, and they work together. One of them is going to help get rid of hydrogens and release them in solution, or release from themselves and put hydrogens into solution. The other one, bicarb, is going to help be able to pull hydrogens out of solution. Realize that if you pull hydrogens out of solution and lock them up, you are making it less acidic, right? You are increasing the pH. If you take hydrogens and release them and put them back into solution, you are making it more acidic. You are decreasing the pH. That's what you got to get out of this. Okay?
So the carbonic acid makes it more acidic and the bicarbonate makes it less acidic.
Yes. See if we can get a better visual on this.
They have to be together.
Yes. Otherwise, you can only do one and not the other. You got to have them both there.
A buffer is an aqueous solution containing both a weak acid and a weak base that are a conjugate acid-base pair. The buffer shown contains the weak acid acetic acid and its conjugate base acetate. These species are dissolved in water. Water molecules are not shown in this animation to draw focus to the buffer components. A buffer solution has the ability to resist changes in pH upon the addition of small amounts of either acid or base. Choose to add a strong acid or a strong base to the buffer by clicking one of the buttons.
Okay, so what we're going to start here is let's look at a buffer system. This is different than the one in the body, but it works. Instead of saying carbonic acid and bicarbonate, we're going to use acetic acid and acetate. Okay, so it's the same principle though. We're starting off with roughly equal amounts of each. Here they are floating around. Notice these guys right here. It gives you your key. This guy right here is the purple with the white dot is the acetic acid. The acetate are the larger green ones. Now what's going to happen is, think about this. If I add a strong acid, what am I putting in? Hydrogens. I'm putting in hydrogens. of what you see there, what will hydrogen react with? The acetate. That's right. Okay? It'll buffer it. It'll absorb any hydrogens that are added up to a certain point. I mean, if you add enough, it's going to overflow. It's not going to work. It has a certain capacity. It's called a buffering capacity. But small variations it can tolerate. If I add a strong base, I'm going to put in hydroxide ions. Okay? If I put in hydroxide ions, what's it going to react with? It's actually going to react with this acetic acid. Okay? It's going to pull this, pull the hydrogens that are floating around as part of acetic acid. It's going to take that hydrogen that it would give off and grab onto it and take the H, hook it with an OH and make what? H2O, water. It's going to neutralize it. Okay? So a strong base is something that's going to be putting in hydroxides. You can do that, there's a lot of alkaline drugs like lithium and a lot of other things that can actually cause, or if you drink Drano, I don't know. These are all can, affect hydroxide ion concentrations in your body. If you put that in, it can resist a basic change. If you put this in, this can resist an acidic change based upon the two things that are present. So let's take a look. We'll add acid first.
Only hydrogen ions, H+ from the hydrochloric acid, are shown entering the solution. Chloride is not shown because it is a spectator ion. It does not participate in the reaction. The strong acid H+ reacts with the base component of the buffer acetate, CH3COO-, reducing the acid component of the buffer acetic acid, CH3COOH. The buffer has resisted a change in pH by removing strong acid H+ from the solution.
So the key is you don't see any hydrogen ions right here floating around anywhere. And if you do, it may be only one or two compared to all the ones that came in. It has the ability, this guy right here, has the ability to hook up with, now look what happens, look at the chemical formula here. If these enter and they hook on to this, you form this. You actually create more of these in the solution. You've buffered that solution. You've absorbed it. You converted the chemical, but you've absorbed it. Okay? If you want to go back the other way, you could actually, cause this to release and you're back to where you were. You could take this, pull this guy off, and you got this. That's that guy you pulled off, and you got this behind. Okay? This is CH3COO-. The green dot would be the purple after that's been separated. Okay? So you can go back and forth. Notice what's happened up here. The hydrogens, when you first watched that, if you were watching this graph, these two were the same, and that kept climbing. And as that kept climbing, what you would see is this dropped. The concentration of this dropped because it was being used. As this was going up, this was dropping because they started reacting with one another. And then at the end, what happens is when they start combining, you make more of this. So that shoots up. Okay? Let's look and see what happens when you add hydroxide ions. If you give it some base.
Only hydroxide ions, OH-, from the strong base sodium hydroxide are shown entering the solution. Sodium ions are not shown because they are spectator ions. They do not participate in the reaction. The strong base hydroxide, OH-, reacts with the acid component of the buffer, acetic acid, CH3COOH, reducing the base component of the buffer acetate, CH3COO-, and water. This buffer has resisted a change in pH by removing the strong base hydroxide from the solution.
Okay, so what happened was this guy, the last time we had hydrogen that hooked onto this guy to make that guy. This time we got hydroxides hooking onto this guy, and look, we've got an H, we've got an H, we got an O. Okay, so this H and this O comes over here, hooks onto that H, and it pulls it off, and what do you got? Water. Okay, what do you got left is this without an H, which is this. Okay, so we effectively bounce back and forth between these two things, and you pick up and release hydrogen hydroxide as necessary. Okay, that's what a buffer system does. Okay, yeah. Yes, it can, but to a degree that is beyond what we're going to talk about. But the temperature will make a difference in the dissociation of that acid. Like in carbonic acid, or in that case acetic acid, it'll actually dissociate more at a higher temperature, in which case you could get increased buffering capacity and that kind of thing. So it just depends. But by and large, we're dealing, of course, with internal body temperature that remains relatively constant, so we don't discuss those aspects of it. But it's a fair question. Okay. All right. So, knowing what we know then, that bicarb has the ability to absorb hydrogens. If you have increased plasma hydrogens, or if you have too much CO2, realize they're both going to lead to the same thing. More CO2 is going to mean more reaction with water to produce more carbonic acid. What will happen is you're going to increase the amount of hydrogen secretion that occurs. So you're actually going to put more of it into the kidneys. You're going to pull more hydrogen out of the bloodstream to help alleviate this problem because there's too much. Which means if you secrete more from the bloodstream into the nephron tubules, obviously you're going to excrete more out of the kidneys. So these two are related. And the result of that, obviously, will be to drop the plasma hydrogen concentration. At the same time, simultaneously, what is happening is your body is signaled to conserve more bicarb. Why? Because any hydrogen that's not getting pulled out over here can get neutralized by more bicarb floating around inside the body. So you want to conserve it, so that means you excrete less of it, and you increase the amount of it in your plasma. So now that it is circulating around, absorbing hydrogens, okay, trying to pull it out of solution to increase the pH, pull it away from acidic back towards where it needs to be. Okay, now this takes us to a very important equation, at least in terms of chemistry. You guys will probably never use it again, but it's the Henderson-Hasselbalch equation. The Henderson-Hasselbalch equation is an equation that helps us calculate the pH of something whenever buffer systems are involved. Different buffer systems, behave a little bit differently. All of them have the ability to absorb and release hydrogens, but each one of them can do that at a little bit different degree. Some of them can absorb better, some of them can release better. There's a lot that plays into that. So just because you add a certain number of an acid, if it was unbuffered, it would be easy to calculate its pH. If it's buffered, the pH is going to be determined by any of those hydrogens now that didn't get taken up by the buffer system. So there's got to be some sort of numerical representation of the ability of your buffer system by which you can use to then figure out how many leftover hydrogens will be there. It's probably not going to be able to deal with every hydrogen that it comes in contact with. So how many of them can it deal with? And this is what the Henderson-Hasselbalch equation will help us figure out, so that we can determine the pH, even though we've got a buffer system present there. Because you can still affect the pH. You dump a lot of hydrogens in there, you're probably going to change the pH a little bit. But the goal of the buffer system is not to remain perfectly stubborn and never change the pH at all. It's just to only allow it to change a very slight amount so that our body can deal with it. Okay? So you will get a little bit of a pH change. To what degree is determined by this particular equation. Now you're going to see a couple things here. pH, we know what that is. I mentioned log a while ago. See something called pK. Not that you need to know, but pK, it's actually pKa. It's just not written, I don't know if it's in your book that way or not. But in chemistry, it's pKa. It stands for the acid dissociation constant. All you need to know about that. First of all, really, the only thing you need to know is how to do the math whenever it's given to you. But, yes, I'll give you pKa values. Okay, and all you got to do is plug it in. That's it. Now, what it represents, just so you know, the pKa value represents how well your weak acid dissociates. Something that has a high pKa value means that it's a weak acid, but it's stronger than the other weak acids that are out there. It actually dissociates better than the other ones. If it has a very low pKa value, then it stays together a lot more and hardly at all gives off any of its hydrogens. Okay, so it's all relative. All the weak acids are relative to one another. Some are stronger than others, even though they're not technically still a strong acid. So the pK value is going to be that, how well your weak acid dissociates. And then what you want to do is add that value. They're textbook values that you can look up. I'll give them to you. Okay? You'll add to that value the log of whatever this value is, which is the bicarbonate ion concentration divided by the carbonic acid concentration. So if you're given a situation where you know the concentration, the average concentration of someone's bicarbonate ions, and you know the average concentration of the carbonic acid molecules that are present, all you got to do is divide those two, put the right one on top, okay, the bicarb's on top, divide those two, take the log of it, add it to whatever pK value I give for carbonic acid, okay, that's your weak acid here, and you'll get the pH for that particular situation. All that you have to be able to do is plug in the numbers. I'll have you calculate pH. I will give you ratio concentrations. I'll explain that in a second. And then I'll give you pK values that you need. Okay? In this case, it'll just be the pK for a carbonic acid, and I'll tell you what that is. Right, but I'll give you that, though, because what you'll see is that here in a second we're going to talk about you don't have to go in and measure the exact number of bicarb versus carbonic acid molecules that are present. What you need is a ratio. Is it a 20-to-1 comparison? Is it a 40-to-1? Is it a 10-to-2? What is it? Because as long as you know the ratio of how much of one you have to the other, you've got this answer. Okay? If you've got 5 times as much, even if you don't know what the value is, you can figure out that you have 5 times as much bicarb as you do carbonic acid, then you know your ratio is 5-to-1, right? So 5 divided by 1 is 5, and you take the log of 5, and then plus the pK value. That's what you would have to do. What you will see is on the exam is I'll discuss, like I'm going to a little bit later, I'm going to discuss certain conditions that are associated with these values, the ratio of these values. You can tell a lot whether a person is experiencing respiratory or metabolic acidosis or respiratory and metabolic alkalosis based upon the ratio of these two things right here. And I'll show you how. Okay? Now, if you can figure out, or you are given a ratio, and I'll give you the ratio in any problem that I ask, that's easy. Like if I say it's a 40-to-1 ratio, then this value right here is 40. 40-to-1. Okay? 40 divided by 1. And then what you'll have out to the side is a bunch of math already done for you. I'll say like the log of 20 is equal to this. The log of 10 is equal to this. The log of 40 is equal to this. And then you pick whatever you need to that applies to this particular equation. In that case, you would say the log of 40. You would see it, and you would use that in your math. Okay? So it's basic addition is what it ends up being, because you'll take the concentrations that are given, the ratio of the concentrations. You'll look up and see what number that applies to that I've given you, the log of what, and you pick which one applies. And then you'll add to it whatever value I tell you is the value for the pK. Okay? So, for example, if the pK for carbonic acid is about 6.1, okay? The log of the ratio of the two concentrations leads to the computation of the pH. That's this thing right here. It says, for example, if the ratio is 20-to-1, if you have 20 times as much bicarb as you do carbonic acid, no matter what the actual numbers are, if you know that it's a 20-to-1 ratio, then you can take 20 divided by 1. It's going to give you the same answer as if you had the actual real measurements. So 20 divided by 1 is 20. Hope you can get that far. So if you know that the ratio is 20-to-1, then the log of 20 is actually 1.3. So I would have, there's common fractions out there that I'll have listed down the side. I'll say the log of this is this, and so on and so forth. And you would say, okay, I'm taking the log of 20. And then you'd look out to the side and see where what I say the log of 20 is equal to. Okay? So it would be 1.3 in this case. So that means all of this now is 1.3. And what you do is add 1.3 to 6.1. What do you get? 7.4, about the average of what the blood is in the body. So normally what this tells us is the normal ratio of bicarb to carbonic acid in the body is about 20-to-1. That's normal. Okay? So what you'll see is, what if bicarb goes up and carbonic acid goes down? What if bicarb goes down and carbonic acid goes up? I'll show you. You can use that to your advantage and figure out what's going to be wrong with this person. Okay? Okay, so all you've got to be able to do is take ratios that I have, look up the log of the value that's associated with it, and the only math you got to do is to add two things together. So certainly I hope that you've made it this far with, and you can add. 6.1 plus 1.3, some of you may need a calculator. I don't know, but take off your shoes if you have to, add them all together. All right. Now, that's a chemical buffer that's floating around. We're still on that first line of defense. We're talking about chemical buffers in the body. The biggest one is the carbonic acid bicarb buffer system. Now, we've also got some other things in there. We've got proteins that are floating around in our body that actually have the ability to buffer a little bit. Hemoglobin, if you think back to that picture I showed you about hemoglobin, it can actually absorb some hydrogen, so it can be a little bit of a buffer. There's some other salts in the body, but the biggie is, there's a lot of them, but the biggie is carbonic acid and bicarb. That's the one you got to focus the most on. Okay? Now, we already know this to be true. Okay? The second line of defense is going to be, in terms of restoring acid levels, is the respiratory system, and it's all about CO2. Here's our equation. Okay? We know that carbon dioxide can react with water to give us carbonic acid. And the second that we get carbonic acid, we know that it ends up being this. It can release into these two things. Okay? And that free hydrogen then is what we have to worry about. Okay? All right. Now, it says here, when hydrogen ions increase in the blood from something that's non-respiratory, if it's a respiratory cause, then obviously the respiratory system is not going to respond. But if it's metabolic, generated hydrogen, whatever, it's something that's not related to the respiratory system can help fix it. The reflex center in the brainstem increases pulmonary ventilation. Why? You increase pulmonary ventilation to basically get rid of carbon dioxide. You want to eliminate more carbon dioxide. I know it says pull to the left, whatever, but bottom line is, if you pull out carbon dioxide, there's less of it to react with this, right? So you're going to get less acid formation. It's going to help reduce those acid levels. Okay? So you want to hyperventilate. In this case, you can increase your ventilation. You can push out more CO2, get it out of the body so that you're forming less acid. And you're either taking down or at least not contributing more to the acid levels that are already present. Okay? Okay. Now, this is going to be an immediate response to the pH change. This is going to kick in very quickly. The respiratory system is a backup. It is not what is most responsible for maintaining this. The most responsible thing is what we've already talked about, that whole bicarb thing. Okay? This returns actually the pH to only about 50%, 50 to 75% of the way back to normal. Okay, so there's still a big gap that has to be filled in. Okay, so obviously as the pH continues to move back to normal, there's less and less hydrogens present, so that means you don't have to blow out as much carbon dioxide. So you start slowing down the breathing a little bit more as the hydrogen levels as they drop. Okay. All right. I was going to say, is there anything else? Because I had one other thing I know I was going to say about it. Doesn't say. It'll come up in a minute then. All right. The last line of defense against pH changes is the kidney. Now, this compensation is not going to be immediate. It's actually going to take hours to days to offset any hydrogen changes that the kidney can work with. Okay, so we depend mostly on chemicals floating around in our body, secondly on our lungs, and lastly on our kidneys, okay, to help breathe things back to normal. We know that it can secrete from the blood vessels into the tubules and vice versa, depending on what is needed. And the nice thing about this is, by the time everything gets to the kidneys, it can work to get pH right back exactly to normal. Okay, this is our final step that can be used to finely tune it and to readjust it right back to where it needs to be. And as a result, urine is usually always acidic. If you check your pHs for the urinalysis that you did, hopefully you would see that it's slightly acidic. It should be. So a lot of hydrogens get pulled out in this last line of defense. Okay? It's naturally something that tends to be a little bit overproduced in our body that we're fighting against, that we have to sort of keep pulling out. Okay. Let's see. That's just common sense. You can get that. All right. One thing to note is wherever hydrogen goes, bicarb is going to probably go in the opposite direction. When we're going back down now here to the renal, back down into the nephron. Here's our tubule. Here's these the tubule cells, basolateral membrane out here, bloodstream over here. So what happens then? If a hydrogen ion is tubular secreted, and in other words, if it's put back into the tubule from the bloodstream, you've got this hydrogen ion that moves in. It says every time a hydrogen ion is tubularly secreted, a bicarb is reabsorbed in the peritubular capillaries. They are coupled. because generally we've got the presence of carbonic acid. So if you're going to pull it apart and put hydrogen out there, you're going to end up getting bicarb back in here. And that's good, because if you're trying to put hydrogens in there, that means you've got too much out here anyways. So kicking this bicarb back out is going to help to reabsorb some of those hydrogens that may be out here. Okay. Let's see. The kidneys also secrete ammonia during acidosis to buffer secreted hydrogen ions. You don't want a high concentration of hydrogen moving through here. Okay. You can also secrete ammonia, which explains a lot in terms of smell and that kind of thing. Ammonia actually can act as a base, and it can absorb hydrogen ions as well. Okay, so that's one thing that can happen out here. And it also talks about some other things that can be secreted in here to help neutralize those hydrogens that are dropped off in there. Okay, all right. Let's see. What does it say? Oh, respiratory versus metabolic acidosis, alkalosis. It depends on, obviously, the cause. Where are these hydrogen ions coming from? Before any compensations, I'm going to explain that in a second. Your body compensates for a lot of things. But if you have a change in pH, and it's because of your respiratory system, the reason that your pH has changed is solely due to carbon dioxide levels. Okay, you're not eliminating enough carbon dioxide. If that is what's increasing your acid levels and decreasing your pH, we call that respiratory acidosis, respiratorily induced acidosis. Okay. Now, we know that in this case right here, we're back to the Henderson-Hasselbalch equation. When the ratio of bicarb to carbonic acid falls below 20 to 1, okay. First of all, how do you get a ratio that falls below 20 to 1? This is interesting too. There's several different ways that we can do this. If you look, I don't know which color is the best, but let's say I have a ratio of 20 to 1. That's equal to 20, right? How can I make that number smaller? How can I make this number right here smaller? There's two different ways I can do it. By changing the numbers that you see there. I know you can't see them very well, but that's 20 over 1. I have a numerator and a denominator that I can play with. What can I do to make the total value of 20 to diminish to a smaller number? Do what? Okay, you're right. I can increase the denominator. If I change it from 1 to 2, I've now went from 20 to 10, right? Now remember what's on bottom. What does the denominator represent? It represents carbonic acid, right? Or I could do what? I could decrease the numerator, right? If I decrease the numerator, I'm also going to get a smaller number. Decreasing the numerator means you're decreasing bicarb, okay? Now, if I want to increase, if I increase the denominator, I'm increasing the amount of carbonic acid present. It could be that I have retained carbon dioxide to make that happen. If I retain carbon dioxide and form more carbonic acid, what I am doing in effect is thereby increasing the denominator. And when I do that, I drop this ratio value. Okay? So as long as you remember what's on top and what's on bottom, that this is bicarb and this is carbonic acid, you can look at the ratio and see what number it is and get an idea as to what could cause that. Either the numerator or the denominator had to change, and which one would have changed, maybe, and how. Did it increase, did it decrease, that kind of thing. Okay, so it says when the ratio of the bicarb over the carbonic falls below 20 to 1, increases the denominator. Okay, the denominator again is carbonic acid. Okay, acidosis exists. You've got more carbonic acid. Normally it's 20 to 1. You want a 20 times as much bicarb as you have carbonic acid. If you increase the denominator, it's not 20 to 1 anymore. Say it's 20 to 2. You've got more acid than what your body is normally happy with. So that means you're obviously going to have an acidosis type of situation. Okay? Now the question is, what caused that extra acid? If it was because of the retention of CO2, then it's respiratory acidosis. Okay? All right. So in this case, you take 6.1, which is the pK, plus the log of the ratio of carbonic acid, of bicarb to carbonic acid. So 6.1, at 20 to 1, we said the log was 1.3, right? If it's less than this, okay, the log of the ratio is actually going to be less than 1.3. When you calculate it, you might see that it's 1.2, 1.1. What happens then? Whenever you have something that's less than that, for example, 6.1 plus 1.1, notice that that's 7.2 now. It only affected the pH a little bit. It didn't drop it all the way down to like 1 or 2, but it dropped it just below where it should be, around 7.3, 7.4. We actually have a little bit of an acidosis type of situation here. The denominator increased. More carbon dioxide was held in, so that means more carbonic acid was able to linger around. That's going to increase the denominator. That's going to decrease this total value. So when you take the log of it, it's going to decrease the log, that total value. So that when you add it to 6.1, your pH is below where it should be. Okay? Takes a little practice. You can look over it again. But all right. Now, what about, okay, in this case it says a lot of metabolic changes, a lot of things can happen in the body that can affect bicarbonate. As you'll learn a little bit later in the next chapter in digestion, we've got organs, or endocrine organs especially, that secrete bicarb ions going out into our duodenum, going into our digestive tract. We've got some bicarb ions that the purpose of those, are the reason they're released is to help neutralize any hydrogens that come from the stomach that are going through the intestine. We have some other places where bicarb can be produced in the body. So it is possible to increase the amount of bicarb that's present in the body. So here's our equation. Let's take a look at respiratory acidosis. Respiratory acidosis has a ratio of less than 20 to 1. How do you get a ratio of less than 20 to 1? In this particular case, we said we can do two things. We can decrease the numerator or increase the denominator. Well, if it's respiratory, it's not going to have anything to do with this. It's going to have to do with this. Okay? So you're going to increase the denominator. Why? Remind me. Why are we increasing the denominator? What's happening? In respiratory acidosis, more acid is forming. Why? Right. You're retaining more CO2, so more carbonic acid is forming. So that's why this number grows. And when this number grows, when you divide those, you end up getting a smaller number. Right? All right, so if you can look at that and see that you're increasing the denominator, then you're having respiratory acidosis. Okay? In respiratory alkalosis, you're going to have a ratio of more than 20 to 1. Okay? Now, how can you do that? You're going to decrease the denominator. In other words, you're blowing out too much CO2. The less CO2 that's present, the less of this that can form. If you drop that number, you drop the denominator, what's that going to do to your total value? It's going to increase it, right? So if you increase that, you end up increasing the total pH. An increase in pH is going towards the alkaline side, going towards the basic side of the pH scale. So since we're dealing with this again still, but we're causing a bigger number instead of a smaller number, we're going to alkalosis. But since it still is carbonic acid dealing with, we're talking about respiratory. Okay? So we've got respiratory acidosis, respiratory alkalosis. Okay, what about metabolic? Because both of these are equally important. Metabolic acidosis has a ratio also of less than 20 to 1. Notice that both of the acidosis conditions give you a ratio of less than 20 to 1. It decreases this total number so that when you add it to the pK, your pH value is lower, which makes sense in terms of acidity. Okay? Again, metabolic acidosis, you're decreasing this ratio, but you're doing it differently. The metabolic has everything to do with the numerator, the bicarb ion, not the carbonic acid from CO2. So if you're going to decrease your ratio of 20 to 1, how are you going to decrease that? Well, if you take this value right here and you drop it and you decrease that value, it's too low, when you calculate your pH, you're going to get a lower ratio here, a lower pH, which stands for acidosis. Okay? So in metabolic acidosis, this number lowers, but because the bicarb lowers. Okay? That's going to be metabolic acidosis. In metabolic alkalosis, this number is going to be too high. It's going to be higher than 20 to 1, higher than 20, because this is actually increasing. The bicarb is increasing. It's too high. So that means your number is going to be too high. When you add it, you're going to get a higher pH, which will be alkalosis. Okay? So it's a matter of looking at this fraction, seeing which one of those are affected. In respiratory, it's always going to be this that's affected, the carbonic acid. In metabolic, it's going to be the bicarb that's affected. Okay? It takes a little thought to reason through this, but you can make the connection if you go back and reread and look over these again. Make sure you look over this slide and you understand why these things are written here. Okay? Okay. Let's see. We already talked about this. We already talked about this. How can you compensate for it? Realize that if carbon dioxide does accumulate, we have a way to help deal with that, of course, and then you rely on the things that are outside of the respiratory system. We rely on those chemical buffers. We rely on the kidneys, since obviously the problem lies with respiration. So we'd have to pick up the slack with the other two things. Same thing with alkalosis, except in the opposite direction. Okay, your alkalosis means there's not enough carbon dioxide. You're blowing it out through hyperventilation. So you're going to have to use chemical buffers to release hydrogen. You're going to have to cause your kidneys to not soak out as much and leave more in the bloodstream to help compensate for this. Okay. So again, if bicarb falls, the numerator, then you're going to get metabolic acidosis. How can this happen? Severe diarrhea. Because if you think about that, a lot of your bicarbonate ions are secreted into your digestive tract to neutralize hydrogens. If a lot of it is secreted and your digestive tract just sort of lets all its contents shoot right through, you're going to lose a lot of bicarb ions when that happens. So severe diarrhea, diabetes mellitus, which is affects the pancreas, first of all, which is going to end up producing bicarb, by the way, also besides insulin. You're going to have a fall in bicarbonate ions as well, because one of the side effects of diabetes mellitus is ketones. We talked about ketones in the urine. The body is not getting, sort of complex, but it's not getting the glucose that it needs inside the cell, so it starts breaking down other things in the body. And one of the other things it starts to break down are fats and proteins. And it doesn't need to, but it thinks it does. And when it starts breaking those things down, it releases little tiny acid molecules and ketone bodies that can turn into acid molecules. And what you get in that is a drastic fall then in your bicarb ions in an attempt to neutralize all of that. And if it's not controlled very well, you can get a pretty good fall in terms of your bicarb ions. Strenuous exercise, uremic acidosis. In other words, too much acidity in the urine. You're actually pulling out too much hydrogens, and that ends up meaning you leave behind bicarb and that kind of thing. So metabolic alkalosis is too much bicarb. If you vomit, for example, you're getting rid of a lot of acid, and that offsets, that leaves behind a whole lot more bicarb. Alkaline drugs, things that are basic in nature, could increase the basicity, the alkalinity of your body. Let's see. What's the same compensations that are in reverse? Okay, chemical buffers, control your respiration, kidneys. All these will work to help move hydrogen where it needs to go. Okay, so we could end on this. This shows you some compensation that can happen in the body. I said that those ratios can get screwed up. The whole 20-to-1 thing. It can get screwed up, but what you can also find, that you'll see this a lot too, like in nursing homes and people that, especially older, that are in this sort of progressing towards renal failure. Some of the things that you will see is some heavy compensation going on in their body to try to maintain these ratios. I'll show you what I mean. This is normal, okay? A 20-to-1 ratio, we'll say, just with blocks, you can get an idea, okay? 20-to-1, when it works out, the pK is about 6.1. The log of 20 is 1.3, so you add that together. Here's normal blood pH. We've got to try to do what we can to keep blood at a normal pH. Okay, so what happens then if something happens and our body doesn't respond, and it's what's called uncompensated? Okay, we don't compensate. If we retain too much carbon dioxide in our body, let's say there's a respiratory dysfunction that impairs breathing that causes us to retain too much CO2 in our body. If that's the case, notice how these scales tip. Where it was balanced was at 20-to-1. I know that doesn't seem right because it doesn't seem like it's even on both sides, but because of the chemistry, realize That's what balances it out. Any change in that tips the scale one way or the other. So even if you add twice, just one more block here, that's twice as much as what was present. Okay? If this goes from 1 to 2, now you have a 20 to 2 ratio. That's uncompensated. There's no changes our body does yet. So how will that make a difference? Well, the log of 20 to 2 is the same thing as 10, right? So the log of 10 is actually 1. So 1 plus 6.1 is 7.1. That's acidosis. That's below our minimum range, okay, for blood. This is respiratory acidosis. Why? Because there's too much CO2. Okay? Now, our body can compensate. If what you can find is if our body retains too much CO2, instead of allowing this scale to tip, we can also produce twice as much bicarb. So what we in effect do is change our ratio now from 20 to 1 to 40 to 2, which is the same value when you divide it. It's the same ratio. It's just we've had to compensate in order to do that. If that increased, that had to increase by the same factor. And it's amazing just how high this can keep going and how long people can survive in these compensated states. Okay? It's obviously a snowball effect. Once it starts, I mean, it's going to compound itself and continue to do that. But people can survive for a while, for sometimes a long while, even though it's really hard on the kidneys in these compensated states. Okay, let's take another look at another one. What if, normally it's 20 to 1. What if we hyperventilate? What if you rid your body of too much carbon dioxide and it's gone, and you reduce this amount? Well, now it's top heavy on that side. You've got a 20 to a 0.5 instead of 20 to 1. But the problem, look at the culprit. Who is to blame for this? Carbon dioxide, not bicarb, right? So since it's carbon dioxide to blame, it's respiratory something. In this case, what is it? Is it respiratory acidosis, respiratory alkalosis? Well, it shows us right here that if this fraction changes, the log of 40 now, 20 divided by 0.5, ends up being 40. Okay, this is times 2, same difference. So the log of 40 is 1.6. So when you add that to 6.1, you get 7.7, which is now alkalosis, which makes sense because we've got less CO2, so less acid forming. Okay? So in this case, it's uncompensated. This is respiratory because the change is over here, but it's alkalosis because the pH is above what it's supposed to be. We can compensate. If we half our CO2, if we blow off too much, then we can stop putting so much of this in our bloodstream, reduce that by half. We can make it a 10 to 0.5 ratio, which is the same thing as 20 to 1. That brings us back to normal. This is a compensation that our body can make. Puts us right back to where we need to be. Okay? Another uncompensation. Now we're shifting. What if the problem is not on this side of the balance, but it's over here with bicarb? It's metabolic something. Okay? Is it metabolic acidosis? Metabolic alkalosis? It depends. Okay? You have to look and see, has it decreased or has it increased? So if you want to, in this case, to answer the questions on the test, you may want to think about these things and just plug in fake numbers like we have here. Something to give you a nice equation that you can come up with. But in this case, if we drop that, we have a 10 to 1 now. Okay? The log of 10 is 1, so we're at 7.1. That's acidosis. Okay? We've tipped it in the other way. Metabolic acidosis. Metabolic because the change occurred with the bicarb ion. To compensate, if I have a decrease in bicarb, but I have a proportional decrease in CO2, I retain the same value of my fraction. So I'm still maintained at neutral, or not neutral, but normal pH blood of the blood. Okay, what if I load up on bicarb? What if my body's producing way too much of it, and I tip the scale that way? Another way to think of this, if you're memorizing these pictures in your head, is if it tilts that way, look at my hand, see? If it tilts this way, it's alkalosis, and you've got acidosis the other way, okay? If you get twice as much bicarb, realize that's going to be a 40 to 1 ratio now. Instead of 20 to 1, plug it in, we get a 7.7 for our pH. It's alkalosis. Respiratory or metabolic? Metabolic, because it's dealing with bicarb. That's what screwed us up. That's the culprit. Okay? Compensated, if you have a little extra here, so long as you can proportionally add a little bit extra here, you can retain the same fraction, maintain proper pH.